Hello, everyone. Welcome to our quarterly ASMDL virtual lecture. We're delighted to have you here, and we're delighted to have our speaker as well. Before we get to the talk, I have a few slides that kind of give an introduction to what this lecture and to what ASM is doing. This program is sponsored by premier program of the ASM called the American Society for Microbiology Distinguished Lecturer Program. This program has been in existence for over 60 years, and it's given hundreds and hundreds of lectures. The idea behind it is that we have a roster of lectures in a variety of topics of microbiology and related that are available to go to the local meetings, smaller meetings where they can really interact with students and anyone actually who's interested in the science, and kind of bring ASM and to the local groups, and to bring the local groups more involved in ASM. So it's been a very successful program. In the last few years, we've actually opened it up because we wanted to reach more people. And so this is why we started this virtual lecture quarterly series for student chapters and ambassadors and students in general. And that's what we're doing today. This program is funded by ASM and a small endowment from the Waxman Foundation for Microbiology. ASM is committed to promoting and advancing science. And one of the other programs that we have that's another program that helps ASM accomplish this goal is a student chapter program. If you are not a member of a local student chapter, we encourage you to contact us, find out what your local chapter is, and join. And if you don't have a local chapter, you're always welcome to start one. And it's very easy process. And speaking of ASM, as I said, ASM is very dedicated to promoting the science and supporting the science and supporting its members as they work in the scientific field. So I encourage you, if you're not a member, to join ASM. But if you are a member, this is the time that you should be renewing your membership. Just log on to ASM website and go through the process and make sure you're all ready to start 2025 as a full member or as a member of ASM. And one of our really, really big events that we're looking forward to is our annual ASM meeting, which we call the ASM Microbe meeting. And this year it will be in Los Angeles. And we are in the process of putting together the meeting program, which means that we put out a call for abstracts. Anyone on this call is welcome to submit an abstract for ASM Microbe. Directions are on the website, and there's just two deadlines that I want to point out. If you are looking to get some money to help with your travel, a travel award deadline is December 3rd. And in that process, you would submit your abstract and then fill out a short form saying that you need and want a travel award. But if you don't need a travel award, or you're not going to apply for a travel award, then you have a little bit of extra time, and your deadline is January 22nd. But more details are on the website, so I encourage you to go there and check it out. So on to our lecture. Today, I'm happy to announce that, or to introduce you to your ASM Distinguished Lecturer. His name is Dr. Andrew Yurochko. He's a professor of microbiology and immunology and the Carol Feist Endowed Chair of Viral Oncology at the Louisiana State University Health in Shreveport, Louisiana. He'll be speaking on understanding the complexities of HCMV entry. And with no further ado, I will turn this over to Dr. Yurochko. And you can share your screen when you're ready.
All right.
Okay.
All right. Well, I want to thank everyone for attending. I want to thank ASM for inviting me, and for the people that supported my ASMDL application when that went in several years ago. I honestly don't know who did that, and I very appreciate that. So today, what I want to talk about is some of the work that I've been doing since I was, I guess, a postdoctoral fellow, and it continues to this day. But I wanted to talk a little about is how I took my field, which I started as a graduate student in immunology, and then continued as a postdoc at UNC. I guess I was a grad student at Virginia Tech, so I got to give a shout out to the Hokies. And then I went and did a postdoctoral fellowship in immunology at UNC and a virology postdoctoral fellowship where I sort of became a viral immunology. And as I've been following the science, we've really become a lot of cell biologists. And I sort of, talk a little about today when I mentioned sort of why I got into science, about following your dream, and in some cases in science, following the science. So I've sort of went from an immunologist to an immunoviral cell biologist. And that's what I want to try to talk about that story today. You heard about the Distinguished Lecturer Program from Anne, but I want to tell you what that really means to me. So for me, when I gave my very first talk, I must have been a second or third year graduate student, was at an ASM talk in Virginia. So really my first professional talk. And although there was the nervousness of the talk and talking about your science to colleagues, what it allowed me to do was to talk to people, which I sort of pointed out. Oh, networking. Can you see my pointer? Does it move? Okay. Yeah. Okay. And I bring
Yes.
that up as sort of, for me, first professional talk, but also was networking. It was where at that talk I met some people in the state of Virginia and those that came to the local meeting that I eventually interviewed with as part of my postdoctoral fellowship. So in that sense, I made my next career choice by meeting some of these people, and it is those people I interviewed with. I've tried to help out ASM. I've been the former South Central branch, which is Louisiana, Mississippi, and Arkansas branch, the South Central branch. I've been the president and now serve as a treasurer. I have gotten or received advice from people within the branch, and I hopefully can give advice to the younger, the trainees. And so for me, I sort of feel like it's my turn to return the favor, sort of pass it forward. And hopefully that's what we're doing. But before I really tell you about my work, I wanted to tell you how I got into science, and maybe a little bit of sort of, like I mentioned in the beginning, follow your dreams or follow the science. So I wanted to be a marine biologist. So whatever reason for me, I always wanted to be a biologist of some sort. And I know my kids tell me I'm 100 years old, but I can assure you that's not true. And they actually had TVs when I was a child, and they even had color TVs. And I remember every Sunday, I think it was, eating pizza we made by hand with the family. We always watched National Geographic. And at the time, the big thing was all the Jacques Cousteau and all the sea exploration of the sea. And it would just seem like a really cool thing to be an explorer, to be a scientist and see things that are new. And I will say it's the nerdy thing of science is when you publish a paper, no matter what, you are the first person to have done that. And you never know in science what that one thing is that you do one day might be in a textbook somewhere, even though at the time you think it might just be trying to get your paper done. So for me, I wanted to be a marine biologist. And although I ended up not being a marine biologist, I at least got to be certified as a scuba diver. And here's me many years ago before I had kids in Australia in the Great Barrier Reef. So I followed one dream. How I got to be a microbiologist, well, that's the point here. My grandfather would tell me about his nephew, my uncle, who worked on the polio vaccine, and thus viruses, immunology, and that sort of thing. And I guess that influenced more than I probably understood at the time, because that's in fact what I did. And I bring that up a little bit more, and I'll show you this picture, is when I was teaching a lecture as a faculty member here at LSU on polio viruses, and I was looking for info on the vaccine, and I found the 50th anniversary picture that was that the University of Pittsburgh did, where they're celebrating the 50th anniversary of the polio vaccine. Of all things, on the front cover, you can see the fellow right here. That's Jonas Salk. But as the arrow points to, that's my uncle, Francis Yurochko, who in that particular moment in time was in the picture that's, sort of preserved in eternity here as part of that. So maybe that influenced me more than I thought. So anyway, here's the things that I think, or at least for me, that I found to be important as a scientist, and I hopefully instill those in the people that train with me, is be curious or inquisitive. And I feel that that was something I loved watching, the Jacques Cousteau and the other science programs. It was fun. It was neat. And you have to have fun, be willing to learn. And I think ultimately, if you do that, you'll find your passion, whether that passion is research, and there's different types from lab, clinical field, teaching, education, policy, science, law, et cetera. All these are things you can do with a PhD. And as I'll mention on the next slide, I have trainees who've gone in, I think almost every single one of those. I also think it's important to understand your strengths and what you like to do. If you like sitting at a microscope for 10 hours a day, do it. If you hate it, do not do that. And that doesn't mean you can't be a good scientist. It means find what you like doing and find a way to do that. And I will say, before I get into what I'm going to talk today, I do want to, before I really get started, is thank all the trainees over the years that have worked with me, and to try to tell you that their excitement, their enthusiasm, their love for science is what really has made my job rewarding. And so, I think you may not appreciate this, but the trainees influence the mentor as much as the mentor sort of influences the trainees. And I think it's important, and I sort of joked in the last part there, science is infectious for all parties. And I think it ends up being a circle that you hopefully instill enthusiasm and a love for that science, and they do that in return, which keeps you going. So that stated, let me talk a little about what I work on. I work on human cytomegalovirus. It's a herpes virus. It does persist for life. So once you get it, you do get it for life, like the other 9 human herpes. There's 9 human herpes viruses, so the other 8, they all persist for life. In the case of CMV, about 40 to 70% of the US population is infected, and the similar numbers are seen in Western Europe. In some place in the world, it is really endemic, where numbers are close to 100%. And what will be important as I talk to you a little bit more about this virus and the work we do is this particular herpes virus is essentially spread in all body fluids, of which I've listed a few here. And that tells us something different about its ability to spread, where the other herpes viruses have a more limited distribution. It just makes them different, doesn't make one better or not, but it does give you some clues to the nature by which that particular virus spreads. And so before I tell you a little about our work, at least would like to put in context the diseases that are associated with this virus. So tell you about why, in fact, we saw this virus. So a big key is it's the #1 infectious cause of congenital CNS damage in the US, and a leading cause of congenital deafness. It's what's considered one of the torch agents or torches agents. It stands for toxoplasma, rubella, cytomegalovirus, herpes, and sometimes they put syphilis in there. These are all agents, whether bacteria or viruses, that can cause congenital infection, meaning an in utero infection affecting the developing embryo. Sometimes we consider a silent epidemic because the mother, unfortunately, does not always know she's been infected, and therefore carrying a potential child who might have a congenital infection. And when you look at the numbers here, it's between 1 in 150 live births and 1 in 200 live births in the United States. In some areas of the world, that could be up to 8%. So we're talking a significant number of live births can be associated with CMV congenital infection. These are some pictures of the little babies that are born. You can see this classic petechial rash. Some of the little babies are, relatively speaking, healthier. Others, sadly, are a lot less healthy. And there can be a whole range of very serious conditions associated with congenital infection. 2 of which are the more serious ones I'm showing here. The first one in this panel A is a little baby born with microcephaly. You can see the brain is and therefore the skull is malformed. They do not have a full brain function, and sadly, these children are born with severe issues because of the lack of brain formation. And in many ways, the same idea of congenital infection, what was seen when Zika virus entered the Americas in the last few years. I guess it was the 2017, 2018 range, somewhere in there. Maybe a little earlier than that. But when Zika virus hit the Americas, we saw a lot of congenital infections in South America. And you can see in the B panel, what I'm showing you here with the pointer pointing to is calcified lesions. These white spots are not supposed to be there in the brain. That's damage. And this particular one, this tomographic picture showing the calcified lesions around the ventricles. I'm sort of highlighting the ventricles that are in this sort of dark figure, is showing issues, and some of them almost show the entire ventricles have serious calcified lesions. And again, that is sort of some of the sad outcomes of this congenital infection. So it's a very serious issue and one that affects a large number of children within the United States. Some of the issues come down to is what I'm showing you on this slide. To your left is if you look at the numbers, you can see somewhere with long-term medical conditions, about 5,500 children are born each year. This is higher than fetal alcohol syndrome, Down syndrome. And so it's a significant issue. And I'll point to this bottom one of about 10 is rubella. So my kids will tell me since I was 100 years old, that I was around during the rubella scourge in the 60s when there was a lot of public health service campaigns to get vaccinated with the measles, mumps, rubella. And you can see how it went from close to 5,000 serious cases a year to around 10 because of vaccination. But here we do not see vaccination yet, unfortunately, for CMV. The reason I point this out, that this is a significant issue, but if you look on your right that I'm circling here, when they asked pregnant women, and these are the percentages of those women who knew about these different syndromes, you'll see on this, the one they knew least about was CMV, yet it was the one that probably most likely or could most likely affect them as far as having issues with the child upon birth. And so this is an education issue. And again, the reason I bring that up is that just a simple hand washing, telling the moms to wash their hands to try to avoid some contact with existing children, can minimize congenital infection spread to the mother during her pregnancy by sometimes over 50%. So it's a significant issue to get this sort of information out there. In addition to the role in congenital infection, CMV is associated with a number of other conditions. The first one I show here is in AIDS patients, and certainly I'm aware that with heart therapy, this has been minimized, but in a few patients that people don't take the therapy, you can still get issues, and I'm just showing you one example here. This is a retina. I don't know if you ever looked at it, but what I'm pointing to right now is this light reddish color that's all uniform is what your retina should look like. These big red blobs are hemorrhaging, and the whiter spots that I'm pointing to now is necrotic tissue. So this is what happens, and obviously there are other issues. And even despite heart therapy, CMV infection in these patients remains an independent risk factor, at least at some level for these patients. Where there is a big issue and where we actually have funding on is in regards to either bone marrow or solid organ transplant, where CMV is looked for as far as a risk factor in bone marrow transplant. The highest stratified risk factor is recipient, so the person receiving the bone marrow being seropositive is a high risk factor. And for solid organ transplant, the highest risk factor is the donor organ, that person giving the donor organ being seropositive. So it remains an issue. And in some cases, which is what I'm showing you here in this picture, is pneumonia associated with CMV infection in a bone marrow transplant patient can have very high mortality if not treated. So like I said, it remains a problem. In immunocompetent individuals, there's also issues, and it causes a minor amount of mononucleosis. For most of you who have heard of mono or think about it, EBV, a related herpes virus, causes the vast majority of mono cases. CMV as well as HSV do cause some cases of mono. There's been a big push to understand CMV and its role in cardiovascular diseases, where it's clear that there's strong evidence in atherosclerosis, restenosis, and certainly in transplant vascular sclerosis, and in animal models, we certainly know they've used Koch's postulates to show, in fact, the rodent viruses do, in fact, or are, in fact, associated with these cardiovascular diseases. There remains studies with linking this to certain types of cancers. Certainly, depending on who you talk to, some will argue with more vigor that is the case, others perhaps with less vigor. But I think when one thinks about the other herpes viruses, there are some that are associated with oncogenesis, so at least it's plausible in the very pragmatic sense for it to be associated with that. And there's certainly some interesting data suggesting that that those that are seropositive, at least late in life, may have a diminished lifespan. So what is the work in my lab? What do we study? We've taken advantage, or what we look into is sort of the hallmark of this virus, and that it's a myelotropic virus. meaning that it infects monocytes and macrophages, and I have them bolded for you to let me know, to tell you that this is what's the topic of my talk. I'm going to talk about how CMV infects these immune cells. And these infection of these cells is really essential for viral spread and lifelong persistence. And what our lab is hoping to do is to really understand these mechanisms of viral infection of these peripheral blood cells and to try to put the idea of this viral biology in the context of disease. So in essence, you might watch the show Criminal Minds, and I know the virus isn't a sentient thing, but in essence, try to get inside the virus, understand why it caused disease, what are the gene products, what is sort of the molecular mechanism by which this virus requires to survive, and therefore that's likely to be behind some of the diseases it's associated with. And that's really what we want to do is understand the processes required, in our case, for dissemination and persistence. Dissemination, I'm defining as spread within the host and ultimately spread from host to host. And then persistence being that once you get this virus, you get it for life, and hoping this provides us clues to disease progression and possible targets for therapeutic intervention. Why have we focused on dissemination and persistence? Well, the real key is viral survival. If you think about a virus, we as the human, in this case as a human virus, are its environment, a bird or a bear or fish, just as three examples. The earth is its environment. In this case, we are the host. The virus requires us, so it has to survive within us. You've probably never thought about this, but from a virus standpoint, your organs are nothing more than portals of viral exit, right? An organ that makes a fluid is a way the virus can spread from host to host. So seeding of organs becomes essential for viral survival. And then this ability to establish persistence. So in the case of all the herpes viruses, once you get them for life. And so you can spread it from the time you get it to the time for essentially your whole life. You can be a potential host to spread from host to host. And ultimately, the mechanism by which it uses to survive and persist is what's linked to the disease that I mentioned to you previously in the immunocompromised as well as the immunocompetent hosts. This is a little model we made many years ago talking about essentially what I'm going to talk about today and really what this virus does. As I mentioned, it's spread in nearly all body fluids. That gives you a clue that about this virus. It's very widespread in its ability to disseminate within a host. It can come, as I mentioned, it's generally sort of thought of as a salivary gland virus, but it is spread in urine, it's spread in breast milk, it's spread in GI discharge. It is a sexually transmitted agent, so it comes in many body fluids. Generally, you get infection of either oral or genital epithelial cells. From there, similarly to EBV, you get infection of a leukocyte. In the case of this virus, you get infection of monocytes. Certainly, there are many questions remaining about the process where I'm sort of highlighting right now. What we do know is monocytes get the virus. They're then essential for hematogenous dissemination of the virus, meaning spread from virus to organ tissue. And it's really this red arrow, which is what my lab has been studying over the many years that we've been doing this work. And in this organ tissue is where the virus persists. And so we know that the virus can persist long-term in congenitally infected children, for example. The virus can be found in urine for months to years. In women post-birth who choose to breastfeed, you can find virus in breast milk for, again, as long as that mother chooses to feed her child with breast milk. So it tells us that this is long-term persistence in those organs, and it does so in the absence of really causing disease in a healthy individual, at least in those organs. From there, these organs, as I sort of highlighted, serve as portals for viral egress. For example, the kidneys, you have tubular epithelial cells that then are important for getting urine to the ureter, to the bladder, to the urethra. Salivary glands make saliva, obviously for spread from host to host in both these cases. From a standpoint of persistence, as the red arrow that just popped up, the virus also makes its way to the bone marrow. And you can see with the red arrow that I'm sort of highlighting, there's a circular route to keep this going for as long as that host lives, allowing sort of this bigger picture circle to occur over the life of that host. So before I tell you what I want to talk, some of the new work we've talked about, I do want to tell you some of sort of the more immunological studies we did, at least to get you a little feeling for what we've done. And it's funny because I look at this slide as I'm telling you this and realize on there, there's data from something like 20 years worth of work summarized on one slide. But really, what we did in the early stage of my lab when I went as an independent investigator is showed that monocytes are really Trojan horses for spread of the virus from blood to organ tissue. And just like in Troy with the story of the Trojan horse, this is exactly what happens. The virus is taken up in monocytes. It cannot replicate in these cells. It moves to organ tissue, and within that organ tissue, it replicates. And we've looked at the immunology of movement out of the blood. We've looked at changes in these cells that allow the ability of a short-lived monocyte to become a long-lived macrophage, and ultimately to create this macrophage and organ tissue that can live or last for months or years, making virus over that period of time. And in essence, everything I've just talked to you on those two slides summarizing 10 or 15 years of work can be shown in this silly little cartoon I've made. And what we have is, what is a monocyte? A monocyte is a cell that lives in the blood and can enter organ tissue. It's important for tissue repair. Have you ever thought about it? If you have damaged skin, monocytes can come in and remove those damaged cells, engulfs those cells, cleans up the damage, allowing replication, new epithelial tissue, and repair of your damage. They also are excellent immune cells. They can present antigen, they can gobble up bacteria and viruses and do things that you can imagine they do, from with phagocytosis and then destruction of that pathogen. So in many cases, I've always thought of this as monocytes being heavily armed janitors, where they have a maintenance and an immunology. What the virus essentially does is, representing this little red dot in there, is it twists the immune response, forcing it to more down the immunological route. It creates unique types of macrophages. And again, I unfortunately don't have time to tell you about some of this old work. And everything it does to manipulate the cells promotes dissemination from motility, survival, differentiation, and lastly, which I am going to talk about today, is really the virology and cell biology of how does a virus enter these cells, forcing a productive infection. And at the end, I'll try to tie it back to disease. Now, we've done a lot of omics profiling. We've transcriptomics and proteomics to identify some of the players. And if you have any questions about that, I'm glad we'll talk about that. But that's not what I'm going to talk about today. What I'm really going to focus on today is, what's the highlighted one, is how does the virus trigger the cellular change that regulate monocyte biology. And I sort of wanted to pick this topic because I found it was interesting when I thought about it, that as a graduate student at Virginia Tech, I worked on integrins. I went to do a postdoc, and of all things, worked on human monocytes, how integrins were important for signaling in monocytes. And then when I switched to CMV, it turns out that the virus uses integrins, in fact, the same beta-1 integrins I studied for much of my career, as an entry receptor. So I thought it was interesting to talk about my change in science from immunology to virology to cell biology, all based on the fact I studied the same receptor over all these years. And what we hypothesized to really put forth how the triggering occurs, or what does a virus do to stimulate these cells, is that there's a unique combination of HCMV-mediated receptor-ligand interactions that trigger distinct and specific changes in monocytes to promote viral persistence. And really what that's shown is on this cartoon here. And here's the virus, and the virus engages receptors. And in many ways, when I came into this field, not 100 years ago, but it's certainly more than 20 years ago now, I approached this as an immunologist, where what we learned was viruses are bad, immune cells are good. And I always thought the virus is nothing more than a giant cytokine binding receptors to activate the cell. And when I started that work many years ago, it was very controversial. Virologists did not want to think that viruses do that. As it turns out, that's in fact what viruses do. And we were one of the early labs, certainly not the first one to identify this, but certainly one of the early ones to pioneer a lot of this work, showing that the viruses through various glycoproteins, and what this is represented on the outside of this virus. These are proteins that are glycosylated on the outside. These are the things that the virus uses to attach to receptors. And please don't get too carried away with what these glycoproteins are. Virologists are not very exciting when they label or decide to call the gene product something, and all they did is took the left side of the genome, annotated to right, and said anything that looked like a glycoprotein, they gave it a letter of the alphabet, GA, GB, GC, GD, GE, etc. Some of them later turned out not to be glycoproteins. Some of them turned out to be, so in this case, the first one is glycoprotein B. We did a lot of the early work showing it binds to EGFR, and that's going to be a critical element. The other one is a pentameric complex made up of multiple glycoproteins, and it binds integrins. And it's really this one that I'm going to spend a little bit more time talking about today. But the idea is that the virus binds to these glycoproteins to cellular receptors, stimulating signaling that make changes in the cell that promote viral infection. Some of them are the cellular activation. Some of them are the viral entry and productive infection. As I bolded here, this is what I want to talk about today. I just want to reemphasize what my point is, that when the virus binds to these receptors, what it's doing, it's promoting or making the cell amenable to viral entry. It's changing aspects of the cell. In the case of monocytes, they're in the blood, which is the right place to pick up the virus, but it's not the right place to replicate the virus, as monocytes cannot replicate. They have to enter organ tissue and become macrophages. And so there's a cell biology and an immunology aspect. And ultimately what this gets is productive infection in those organs, which allow the seeding of the organs and the spread. So getting back to my model, this is the element I want to talk about. And it's sort of fortunate that when we started this work, I guess I was an assistant professor at the time, we started learning more. They started doing genetics and sequencing the particular virus. What we learned was we'd always been using a clinical isolate that had the correct series of glycoproteins called the pentamer. Again, the pentamer simply being 5 glycoproteins come together. Those 5 glycoproteins that come together are important for binding both beta-1 and beta-3 integrins. A lot of labs used laboratory adapted strains that lost this pentamer, and so they could not bind monocytes. And so at the time we started sequencing, we had these 2 viruses, 2 very different biologies. And what's a cool thing we can do is we basically learned that what was done naturally is we broke the system. And as biologists like to do, you break something and see what happens. You take the natural and you take the broken one and see what happens. And so this is just some very simple experiments were done. And I'm just want you to focus on what's in the red there. And what we have in this case in the A panel, mock, which is monocytes treated identically except with no virus. I have a virus that has all the wild type characteristics, so it has all the appropriate glycoproteins that can infect every cell. And we have a virus that is a mutant virus or a broken virus. And what I'm showing you here is a number of Western blots at 15 minutes, and we're looking at the changes. So you can see, even though the numbers may not appear to be dramatic, I can assure you they are. This is 15 minutes. The numbers continue to go up if we look at 31 hours. It tells you how rapidly there are changes associated by having a clinically relevant virus that can bind appropriately versus a mutant virus. What's the outcome of that is what I'm focusing on here, in which we look at the ability of the virus to enter the cell. You take the virus, we infect the cell, we wait an hour, we strip everything off the outside, we look at what's inside by PCR, and what you can see within an hour, there's about 3 times more virus inside when you have the correct series of glycoproteins, so appropriate signaling, versus when you don't. And I'm going to tell you right now, I'll show you more data, but this will lead to productive. Even though this gets in at a low level, this is not productive. So there's more going on. And what this told us when we started this, in a few more slides, I'm going to show you that there was something very different going on in these cells, and it piqued our curiosity. And by piquing our curiosity, we looked in more detail at what was going on, and I will follow up with what the answer is in just a minute. I'll leave you hanging for just a moment. So what it did tell us is you have to have the right series of proteins to enter the cell correctly, and that correct entering was going to make a productive infection. And I will tell you just some data from a very new study that I'll talk about near the end. This point of signaling when you have the correct series of glycoproteins versus when you do not is absolutely true. This is a phosphoproteomic array in which we looked at something like 300 different phosphorylated proteins. And in black is the correct virus that has all the wild type virus to infect clinical cells such as monocytes. The lighter gray bars is the virus that does not have that. It's the broken virus. And what you can see in each black bar, there's a much higher, or in some cases, a positive signal, while the gray in most cases a negative, except there's one case where you do see a little bit less signaling. And each of the arrows here point to ones in this particular study that we've also confirmed with Western blots, really verifying that there's a complex positive signals when you have the correct series of events. In the absence of that, bad things happen, at least from the virus standpoint. And really where this hit us and led us to more studies that we'll follow up on, as you can see, this is an older study from about 10 years ago. And if you would just focus on the 3 red boxes, starting right here, when we started to look at what happens to the virus when you let it infect these cells, in our case, went out to 5 days. And if you look at just in this insert, I know we faux colored the nucleus in red here, and I hope you can see what I'm pointing to is a green dot represented by the arrows here, but in the blow up, that is a fish staining looking for viral DNA in the nucleus. You can see at 5 days when you have a regular virus that has the correct series of glycoproteins, it can be found in the nucleus. And look up here, same insert, and I know I'm only showing one cells. We looked at hundreds of cells, and you could see no virus. So even though at 1 hour, some of this virus does enter the cells, by 5 days, there's nothing left. This was confirmed up here in the D panel in PCR. You can see at 5 days, we can find presence of the viral genome in the virus that are the viruses that have the clinical like combination of glycoproteins and viruses that are broken. As I've seen here, the mutant virus, you see nothing. We go to 15 days, you can now start to see the very first gene products being made. And all IE172 stands for is immediate early 1, the very first protein made, and 72 stands for the kilodalton. This RNA, you can see PCR, you can see the very first message for what will become, which we did do, Western blots look for protein. We can see protein as well in the same time frame. So what this tells us really is that when you have the correct series of glycoproteins, you not only get correct signaling, but you get the ability of the virus to enter the cell. You get the ability of the virus to productively infect and make its way to the nucleus. And I'll tell you a little bit more about why that nuclear entry is so important in just a minute. And so from that data, the next person in the lab, so Maciek graduated, moved on to a company in Boston. Junhong Kim entered my lab, and we were curious about the findings that I showed you on the last slide. So we decided to take a further look or in more detail. As I sort of hinted at, we were curious or inquisitive as to what might be going on. And what you can see, this is very simple, old classic PCR done where we looked at endothelial cells. We've done the same experiments in fibroblasts and epithelial cells, and then down here is in monocytes, and you will see a very different time frame. If we look at every other cell type we looked at, you can find viral DNA in the nucleus. So we infected the cells, we removed the cytoplasm, isolate the nuclei, look for viral DNA. You can find HCMV DNA at 30 minutes, 1 hour, and obviously stays out for the course of infection until the cells lyse. You can see in monocytes, we find nothing at the same time frames. In fact, we didn't find anything at 1 day, 2 days, only starting to see it 3 days. And what this told us, something very, very different was going on. It really piqued our excitement, really got us excited about the data, because I think it told us that something very, very different was going on in these cells and same other cells. And I think it was going to tell us something a lot more about the immunology of monocytes, how the virus in turn has to infect how it has to affect and deal with these different cells, and really gotten a lot of neat, at least we felt, neat cell biology to look into this. So I'm going to give you a little cartoon to sort of tell you what we saw, and then I'll show you some of the confocal pictures, some of the older ones, and then some of the newer ones. So getting back to that cartoon, here's the virus, here's the glycoprotein. Glycoprotein B binds EGFR. The pentamer binds the beta-1 and beta-3 integrins. These things are required to get signaling to enter. What we find is the virus goes in early endosomes. We know there's signaling involved. If you have a defective virus or block the correct signaling through inhibitor drugs, the virus gets in late endosomes and gets degraded. And that's why even though a mutant virus does enter, it does not signal appropriately, it gets degraded, and there's nothing left around 1 day. However, if everything is normal, the virus then makes its way to the trans-Golgi network, to what we consider recycling endosomes, and from there, you get de-envelopment. And I told you I'd remind you why nuclear translocation is so important. The capsid then is released, it docks at nuclear pore, and it extrudes the DNA into the nucleus. And herpes viruses, all the herpes viruses that we know about, from the human herpes virus to the animal herpes viruses, follow the mammalian paradigm, and that they have to replicate, or the DNA is replicated, message is all made off of those transcripts, and all that occurs in the nucleus, meaning it's following a mammalian paradigm. This is different from, say, pox viruses as a DNA virus that replicates in the cytoplasm. So this all has to occur in the nucleus. So this nuclear translocation step really is essential for viral replication and is essential for all the subsequent steps of viral infection. So what did we do to sort of get the data that I sort of let the cat out of the bag showing you in that cartoon? Well, the early work that Jun Hong did was an older confocal microscope that we had at the time. This was eventually published in 2016. And what I'm showing you here is we have a GFP labeled virus on a tegument protein. And essentially what this means is the capsid of the virus, when the capsid is enclosing the DNA. So it's going to be following essentially the DNA as it traffics into the cell. And then we can look at cellular markers, early endosomes, a marker of trans-Golgi, a marker of recycling endosomes. I'm only showing you 3 here. We looked at all the other vesicular possible pathways into different vesicles within a cell. These are the 3 that are important. We also looked at other glycoproteins, or something that would be equivalent on the outside of the envelope, so we could traffic the whole virus, then when the virus de-envelops. And we did this from 15 minutes to 4 days. And then what I've got is arrows, in the color of the arrow represents what's occurring. So yellow is co-localization between the green and the red. So here at 15 minutes, you can see that the virus is co-localizing with early endosomes, at 2 hours with the trans-Golgi, at 1 day with recycling endosomes. At 2 hours, the virus is a whole viral particle, completely different. And really, we believe the first time this was reported, where it's a whole virus, hours into the infection, it doesn't occur in other cells. Now you see it 1 day, we're seeing some in which is still there as a whole particle, others where you now start seeing de-envelopment, and by 4 days, you do no longer see any co-localization, meaning the virus is already encoded. And in this case, the capsid and the DNA is usually sitting at somewhere around the nuclear pore, and then the glycoproteins are just milling about and eventually be degraded. What I want to put that in the context, we quantitated everything through multiple different mechanisms. This is the one via the microscopy. And what you can see is everything I've just stated here. If you follow my cursor, this is the ability of the glycoprotein to track with the capsid or the DNA. And you can see for the first day, it's a live viral, a whole viral particle is still in there, meaning that the whole particle, infectious particle, is retained for up to 1 day. And you can see the numbers go down when it's near 0, right when you see all the viral DNA in the nucleus, or start to see viral DNA entering the nucleus. You can see the virus starts out in early endosomes for about the first hour. You can see that starts to now see a peak in which it's in the trans-Golgi network. And then that goes down, and now you can see the virus really sorry about that, is staying in recycling endosomes, and that then starts to go down through 4 or 5 days when we see this sort of the peak, the initiation of the viral DNA into the nucleus, and it continues where it's already peaked at 5 or 6, and not much helps happens really after 7 days. So it's really something very interesting, delayed trafficking. That got us excited about cell biology. We talked to some cell biology folks to give us more clues on this. And I just want you to focus in really on these red boxes and really at what I'm highlighting here. And they said, well, if this pathway that I'm highlighting here with the cursor, which is the arrows, is important, you should be able to break something and prevent that infection. There's really not any way to deal with this, at least not easily. We found this to be this step to be rather difficult. There are a lot of nice inhibitors and drugs that other people have used in the cell biology field to block this, and that's what we did. And what you can see here, if you use a drug that blocks the trans-Golgi, when we block it, we no longer see virus in the trans-Golgi. If you don't use a drug just to control, you see plenty of virus at 1 hour in the trans-Golgi. If you break that down in more detail, which is what I'm showing you here, and what we're looking at is the 3 different areas of cell structures: early endosomes, the trans-Golgi, and late endosomes. And I'll walk you through the slide. If you look at DMSO, which is the control-treated cells at 1 hour, the virus is not in early endosomes, and it all can be found in the trans-Golgi, which is right here, which is exactly what we saw in that quantitative slide I showed you on the last one. However, if you block this step, which is shown with EXO2, all the virus is stuck right here, right? There's a roadblock. It cannot go this way. It is stuck. So you block what I'm highlighting here, it is stuck here. It can no longer follow the pathway. That's in fact exactly what we see. If we follow the experiment longer, if this is broken, this thing cannot go anywhere while the control-treated cells go the normal way, meaning that you have to have this step. It's critical for infection. Why is that? We're still learning some of that, but at least what we noticed in this slide, which gave us some clues and some follow-up studies, is the virus does in fact seem to get inside the trans-Golgi. So this is taken with the time we're doing this study. We got a newer high-resolution microscope. I know it looks like a blue cloud. This is the nucleus. We have stained the trans-Golgi here in red. The virus is stained in green, and then yellow is co-localization. And what we're sort of looking is down on the cell. Nucleus is sort of on top. And what you can see is a yellow in which it's inside the red. As we start taking slices through, I know it's hard to see, but as you start taking slices through that section, which is what the level, the microscope allows us to do, you can start to see the green appearing, meaning that the virus is actually inside the trans-Golgi. And so that it gets inside, we have some data, although I'm not going to show it today. We've identified some of the vesicular transport proteins, the T snares that are associated with this, and there's some unique features of that ability in monocytes, and I wish I had more time to tell you about that, but I do not. But I did want to tell you that the signaling that we talk about infected cells, that it is important. And although this particular one is with the GBEGF, it shows you what we've been able to do is we got the higher resolution microscope, and I'll walk you through this. But I want to point out that every dot or every triangle is an individual viral particle that we individually measured in relation to its ability to find that particle into whatever it's close to. In this case, how close is this virus to the trans-Golgi? How close is this virus to the recycling endosome? And what you can see is when you have virus that's control treated, it's co-localizing with the trans-Golgi, is shown in this red box. And if we look at a later time at 1 day, it also localizes with the recycling endosomes. And this is to remind you where de-envelopment occurs. And if you block the signaling, we see the same thing if you block the integrin signaling, you do not find the virus at all localized to trans-Golgi. And as a net result, it's not in recycling endosomes. And as a net result of that, the virus cannot make its way to the nucleus, and thus it becomes a dead virus, meaning that this really is an important facet. Summarize where we're at. What I'm telling you, what we focus on mostly is that when the virus binds with this pentamer, and I know it looks like a Mickey Mouse here, but these are 5 distinct proteins bind the integrins. This is a positive event resulting in trafficking. There are specific signaling molecules. I didn't talk much about it, but I highlighted it in a few of the slides. Sarc signaling is required, but I mentioned P10 because I'm going to end my talk with the new data just came out this year on how we believe we have identified a single protein that can overcome a mutant virus. And when you don't have this pentamer signaling, you have negative signaling, and in many cases, degradation. So I showed you this early on, I don't know, maybe 10 slides or so ago. A phosphoproteome array, did 300 and some proteins. I only showed you 5 or 6. This is an expanded one. Again, the same 2 viruses. Virus in the gray bar, which is the virus that has the mutation, doesn't have the correct glycoprotein, only can infect fibroblasts, or the virus that has a clinical glycoprotein, so it can affect monocytes, epithelial cells, all the appropriate clinical cells. We use an ELISA-based assay that can look for 1,318 different phosphospecific sites that translates to about 300 distinct phosphorylated proteins. Because remember, some proteins can have 2, 3, or 4 different sites. And we did that again. You can see if you have the correct series of glycoproteins as emphasized in the black bar, we see positive signaling. If you don't, you see negative signaling, except in one case. And I point this out simply because this is EGFR, and the mutant virus still has GB and still does some positive signaling with EGF, but not the same level as the virus that has the clinical. What we noticed from this was this particular protein here, P10. What is P10? P10 is this protein here. It's a phosphatase, and I'll try to explain this in a little bit more detail, how we got into this and why we thought it was important, but I at least want to point out what it does and why it fit. We believe when we found this was we were first a little worried because we didn't know what exactly it did, but as we read more, we got more excited and we got more curious to read more about this particular protein. As we read more, we got more and more excited. So P10 is a phosphatase. It's generally thought, which is shown here in the 1 panel, to take the phosphate off the inositol ring in the 3' position, make it to a 2, to only have 2 phosphates on there from a PIP3 to PIP2. This is what a lot of work has been done on, and it antagonizes a kinase called PI3 kinase. This was done a lot in the literature, and we saw this. We saw P10 go up. We were a little disappointed because this is what was mostly studied in the literature, thinking, well, we just confirmed the literature, which is great. We'll have to go back to drawing board. But as we read more, we realized that this particular molecule has a second function, and it told me and the students and postdoc who was working on this that you have to go in more detail, think more, read more, and we realized it's a dual phosphatase and actually plays a role as a protein phosphatase, looking at these RABs, which control vesicular transport. I don't have a lot of time to tell you about all the RABs and how they control vesicular transport, but that got us excited that here is a RAB that controls vesicular transport, and when the phosphate is taken off, oops, sorry about that, I keep hitting the button. When the phosphate remains, this vesicle that has a RAB7 with a phosphate does not mature. Because it doesn't mature, it doesn't turn into a late endosome and doesn't degrade its material. If you're a virus, you do not want to follow this pathway and get degraded. So what do you want to do? You might want to phosphorylate P10 on an inhibitory residue, and that again was our thing. We saw P10 phosphorylated. At first we were bummed, and then we looked and realized it was phosphorylating every single inhibitory residue on this molecule, which we felt the virus was probably making sure that P10 was turned off to allow this to stay phosphorylated, to allow the vesicle to stay in an immature form, to allow that same transport pathway that we talked about. And I'll show you the data in a minute, just summarizing what I'm talking about. This is required for signaling. The other one is the absence of signaling. So does something happen when we knock out P10? The answer is yes. You need P10 paradoxically for entry, and you need P10 paradoxically for transport. So we knocked out P10. What we noticed in just the red box, your virus, we used an siRNA, so we transfect siRNA in. It gets rid of, as you probably know about how siRNA works, it removes the message P10 goes away, less protein based on the half-life of the protein. And so in the absence of P10, or when the scramble control, we see perfect entry. In the absence of this, we see lack of entry, which was cool, but also interesting because we weren't sure how it might affect trafficking, but it told us that P10 was important. We were able to use a drug and we saw the same effect. So we could knock down P10 molecularly. We could use a drug to get rid of its activity. We saw the same thing, suggesting it's important. I'm not going to have time to tell you about it, but what identified is that when you mess with the membrane before the virus can enter, it requires certain membrane components to cross that membrane, adding to important role that the vesicular transport and the early steps in transport may have. What we later learned was that P10 also plays a role in the later aspects of trafficking in those pathways I've done, what I've shown you in those cartoons. And I'm only going to focus here on the one that I'm highlighting and in the red. So this is looking at 30 minutes post-infection. Here is the virus, in which is a wild type component. It has all the correct glycoproteins. Here's the same virus we treated with SF. This is a drug that inhibits the ability of P10 to function. And what we're looking at is distance, with each of these circles being an individual viral particle that we've looked at in an infected cell and counted it in nanometers, or I'm sorry, microns, to the vesicle of interest. So you can see we're pretty much at the limit of detection for the confocal microscope. We can see the virus is essentially co-localized with early endosomes. Why do we see no effect here? Because the virus turns it off. You can't turn off an off switch with the drug. Now, look at the virus here. This virus here does not have the correct series of glycoproteins. This is the one that gets degraded, doesn't enter right, doesn't stay long in early endosomes because it goes right to late endosomes. Where do we see it? Not really co-localized with early endosomes, not a productive pathway, but an unproductive pathway and gets degraded. But look what happens when we now turn off the pathway. We now see it returns pretty much to just like wild type. And again, we were excited because now we had a mutant virus that can't do correctly. We found a single enzyme that we could shut off and essentially take a virus that's a mutant and make it look like a wild type virus by simply identifying a single player. We could, in fact, start to rescue, which is what I'm showing here, nuclear translocation, because that was the ultimate thing the reviewer asked us to clarify. Okay, great. You can affect each of the transport pathways, but does the virus make it to the nucleus? That's the real hallmark of your pathway. And the answer is yes. You can see here, this is looking at PCR, and essentially little or nothing makes its way to the nucleus. This is really at background. When we treat the virus that ordinarily cannot make its way to the virus, you see a significant increase in the ability of that virus to make its way to the nucleus, ability to start making viral genes and ultimate viral proteins. So we can take a virus that's a null for a specific series of glycoproteins and make it a positive with a single enzyme, or sorry, via manipulating a single enzyme. So what I'm sort of showing you here as we're nearing the end here is really a summary of many, many studies, but highlighting our last paper just came out this spring, is that these glycoproteins, via binding of integrins through Src signaling, phosphorylates P10 on the important residues that blocks this process right here, because you cannot dephosphorylate. This keeps this particular enzyme or this particular RAB, which is associated with vesicular transport, keeps it in the positive standpoint for the virus, negative for the immune cell in that context, allowing the virus to positively transport through the pathway, in essence, to follow this pathway. Here's these glycoproteins, and I wish I had more time, but we've looked at this in detail. We've looked at this one in detail, and I've only shown you a little bit of detail, and I thank all the students who did this work. But these things work in tandem to regulate signaling, to get to early endosomes, the trans-Golgi I showed you is actually inside this. We have a lot of the data that this is a really cool biology of retrograde transport, essentially being transport backwards, require some of the neat processes, these snares, finally to recycling endosomes where you get you get this de-envelopment. And from the standpoint of this virus, it allows the capsid to be released in the cytoplasm, dock to the nuclear pore, and extrude its DNA in to start the process. Have we got some pictures from EM? Yes, you can see here. This is one that I did not use the EM, but these are our cells. I think it might even, it's from donors in our own lab. Very classic macrophage. But if you can look at what I'm pointing out here and blown out here, here's a little finger-like projection, sort of a very classic micropinocytotic vesicle, little finger hooking around, which will eventually close, and then you'll get entry of that virus into a vesicle, which is exactly what we see. And this is one by chance. We took at 30 minutes, really even before we did some of these studies. So we got really lucky, picked the right time point. Here you can see virus at 30 minutes, exactly where it's supposed to be. I wish we could do some more, and that would be a good goal to see what happens at these later time points. So put everything back into perspective. Here is our pathway that I've showed you. This is a virus that's spread in nearly all body fluids. It's very prolific in its ability to spread. It's probably why it has a rather high percentage, maybe not quite as some of the other herpes viruses, but it has a very good transmission from person to person, starting in childhood, either mother to child transmission in daycares, then later in life during the onset of sexual debut. These are really the three stages in which, or the two sort of time frames, early and a little bit later, where you get spread. You get spread in epithelial cells. Again, this is usually a salivary gland virus, although it does spread through genital epithelial cells as well. You get spread to monocytes. This is really where my lab is focused on. And as this double circles are hints, this is what we talked about today. How is this important disease state? This is where we know this infection of monocytes is really essential for infection of neonates and certain vascular diseases where CMV is associated with atherosclerosis, with transplant vascular sclerosis and restenosis, where you do get infected monocytes and macrophages in those sites. You do see spread to organ tissue. We know very clearly that this is associated with solid organ transplant, bone marrow transplant patients, AIDS patients. And what I didn't spend a lot of time with is cancer patients who are undergoing chemotherapy. A lot of times they're immunosuppressed. And in some cases, well, in congenitally infected neonates, you get infection of organ tissue. From there, this organ tissue allows a persistent infection, and then you can reinfect epithelial cells at those sites, whether it's genital, mammary gland tissue, kidney, GI, or salivary gland, to name most of the ones, and then get spread from host to host. Now, I didn't do any of this work. I realized this was an older picture I put together, and I have to go to my lab and get a newer picture, some of the newer students, but these are a year and a half ago, some of the people that did the work. But I want to make sure we talk about Jamal. He's done a lot of the work, and all the older students who are not on there, which I'll list in the next slide, some of my funding sources. But a lot of the work was by what we talked about today was Bailey, who's now a postdoc, or was a postdoc, a graduate student and a postdoc, now doing clinical work in the University of Rochester and Victoria. But Joseph, Jamal did a lot of work. Here's me. This is a high school student, a postdoc, and we have some others in the lab that I unfortunately didn't show. And I'll acknowledge all of the people that over the years have worked this, and this was the fourth or fifth slide, thanking all these people. They made science fun. They made my lab fun. I hope they had fun for me, and I hope they got something out of this. And my funding sources down here. And with that, I'll take any questions.
Thank you so much. Great presentation. I think it's a lot for everyone to deep dive into, but I think it was a great introduction on what the work that your lab's been doing. I just posted, anyone is more than welcome to add any additional questions to the chat. We are up on time, but I do want to make sure we have at least one or two questions that we can address today. Lots of thank yous coming into the chat, so thank you for that. So there's one question here from George Vidal. What are the challenges for therapeutics and vaccines against HCMV?
That's a great question, and I would have wished I had more time. So the challenge with CMV and a vaccine, and the reason it's failed, is you can be infected over and over again. So with CMV, traditionally, when I started in the field, everyone thought of a primary infection, meaning the first time the mom saw the virus is when it was the danger period for congenital infection. But a more recent discovery really is that it's anytime you get infected during pregnancy. So the danger period is 2 months prior to conception to 6 months post-conception, whether it's a primary infection, again, meaning a new virus, or a reinfection, meaning a virus from you've already been infected, you're seropositive, you get a new virus, whether it be from a child or any other source that you might pick it up from. That particular reinfection can have the same exact outcome for the, at least in the context of congenital infection, as a primary infection. Excuse me. So the challenge has been that there's likely hundreds or maybe even thousands of strains of CMV. And we certainly know from STD clinics, people can have dozens and dozens of infections, meaning they're getting infected, reinfected all the time. And each individual one seems to survive within that person. So the challenge has been that this ability to get an infection, a challenge, and then be protected against other strains is difficult. So the reason our work was successful over the years, little backdrop, is we used the very first vaccine strain, is what I used. I got it from my first mentor who got it from the original vaccine trials. And luckily, of all things, that glycoprotein complex called the pentamer was seriously stabilized, where every other clinical isolate that people get out, it's lost in 3 or 4 passages. Ours was stable, allowed me to do all the work in monocytes early infection before I knew about the genetics. That particular vaccine strain, when it was used in patients and then they used it to challenge, it worked great. But because there's dozens and dozens or maybe hundreds, it has not. And the last thing I'll end with is a good comparison, to answer your question, of Zika versus CMV. And these were studies done in Brazil through a number of colleagues. When Zika virus seropositivity went up in the Americas, disease went down, because as the women became seropositive of the virus, they no longer were susceptible to potential congenital infection. With CMV, as seropositivity goes up, disease goes up, because now you have more people shedding virus, different strains. And so you now have somebody who's more able to get that virus from somebody else. And so that is what's really been the hurdle. And a lot of the companies trying to make a vaccine have really struggled with that. Some have even given up. Now, a number are still trying. We're trying to find the magic glycoprotein that gives at least potentially protective immunity during, say, pregnancy, which would be an ideal scenario. But at present, that has chosen to be a real struggle.
Yeah, thank you, Andrew. Amazing talk.
Thank you.
Yes, thank you so much. I want to thank our speaker for the great presentation today, as well as everyone who was able to attend live. And we will be sharing the recording with all of our registered attendees. And if there's any follow-up questions, please feel free to contact ASM at studentchapters@asmusa.org, and we're happy to facilitate connection with our speaker. So thank you all for a wonderful day and evening if you're over here in the US. So thank you.
And if anyone wants to ask me some questions, you can email me and I'll be happy to try to answer them. I know we're out of time for that. And sorry, Jorge, I should have made my screen bigger. I was just reading the things. All right, thank you guys. This was a
Enjoy the
real
rest of your
pleasure.
day. Thank you so much.